Okay, can everyone see my screen okay?
Yes.
Yes, okay. Okay, so again, thank you so much for having me this evening. I'm really excited to be here. And when I was preparing this talk for this evening, I'll tell you, I thought a lot about the picture for the cover slide here. And that's because I thought, oh, I should really have a picture of a big showy flower because this is the Dahlia Society of Ohio. And but you know, I'm not a gardener. I am an ecologist, a microbial ecologist at that. So someone who studies microorganisms and the environment. So I selected this picture because I think it's equally impressive as a showy inflorescence. This is a picture of one of our forests at Holden Arboretum in the springtime. And there's a couple of us in here hiking out to one of our sites. And what you can see is just this dense cover of wildflowers. This is what the forest floor looks like out at Holden from around, early to mid-April into mid-May. These spring ephemeral wildflowers just blanket the forest floor. Most of what you're seeing here is wild leek or ramps, if you've ever had leek and potato soup. And so it really does smell kind of oniony out there. But we actually have a really nice diversity of wildflowers out in the forests. Oops. Here's some of the species of wildflowers that we find. These pictures we all took within the past couple weeks when we were out doing our wildflower surveys. This one is my favorite. I really love Spring Beauty. This one here on the top left. This yellow one here is Trout Lily. This white one here, this is Trillium, white flowered Trillium. We get red flowered Trillium as well. Sometimes that's called Wake Robin. This one here on the bottom left, this is wild geranium. Last week I was out with a high school student who was shadowing me, and it was so pretty to see these little pops of purple all throughout the forest floor. And then we get these two species as well. They're in the same genus Dicentra, but two different species. I can't tell the difference between the leaves. I need to have the flowers to tell that this one here in the middle is called squirrel corn, and this one here on the bottom right is Dutchman's breeches with the pointier flowers. So now I don't study the flowers per se. What I study is the soil beneath the flowers. And so here's some pictures of the lab members collecting soil out in the forest. And this is going to come as no surprise to anyone when I say that the health of the soil dictates the health of the plants growing in it. And this is true in a horticultural setting. This is true in agricultural setting. This is also true in a natural setting. And about, I would say 20 years ago, when I was starting graduate school, I feel like there was this shift in how we think about soil. We went from thinking of it as this non-living medium for plants to get nutrients to something that is very much alive. So this diagram here is of the soil food web. We show this a lot to students that come through the Arboretum on field trips as a way to illustrate that the soil very much is alive. There is a diverse biota that live in the soil, including all of these organisms that we see here. And the ones that I focus on as a microbial ecologist are these two groups, the bacteria and the fungi. I don't want to give bacteria short shrift. They're certainly important. But when we think about nutrients and how they cycle through soil and decomposition, the fungi really are the main players there. And so hence the title of my talk, The Fungi Beneath Our Feet. We really do quite a bit more with fungi than we do with bacteria in the lab. And now these two groups of organisms are very diverse in the soil. There are estimates that in just 1 teaspoon of clean, uncontaminated soil, there can be up to 1 million species of bacteria and fungi. In fact, the most recent estimate I heard for fungi are around 20 million species worldwide. So this is an incredible amount of diversity, and the majority of these species are really beneficial. And of course, we don't always think about microorganisms as beneficial. And in some way, we've done a disservice thinking about microorganisms this way. We think about them as being pathogens. And, I'm not trying to downplay pathogens. We are, of course, in the middle of a pandemic here, and pathogens can cause a lot of harm, of course. But it really has been a little bit detrimental to think of microorganisms only as causing disease. And anyone who's seen The Big Bang Theory, you know that this is Sheldon Cooper in this picture here. He's a huge germaphobe, and anytime one of his colleagues gets sick, he pulls out his can of Lysol and he's spraying the air so that he himself doesn't get sick. And we have this culture of lots of soap and hand sanitizer. I'm always hand sanitizing my kids' hands. But the reality is most microorganisms are beneficial. And so we see this in everyday life. When we eat yogurt that's made by bacteria, or when we bake bread and use active yeast for the bread to rise. And there's a word actually for these beneficial microbes. Maybe you've heard this. It's called the microbiome. The human microbiome are all of the microorganisms that live on and in a human body and are beneficial to us. So we have microbes that line our gut and help us digest our food and cover our skin and help us protect against disease. And this has become such an area of active research that there's a whole journal dedicated to it called Human Microbiome Journal. Now, I, of course, don't study the human microbiome. I study the soil microbiome. And so this, as I mentioned, this is the bacteria and the fungi in the soil. And it might be a little funny to think about fungi as microorganisms or part of a microbiome, because many of them are not unicellular. Some are. Some fungi are really simple unicellular organisms, but some are not. Some are more complex. However, we do lump fungi in with other microorganisms in the soil. And that's for two reasons. First is the methods that we use to study fungi are the same as the methods that we use to study other microorganisms in the soil. And also because the majority of a fungus's life is spent below ground. This diagram here is of the fungal life cycle. And so, we're used to thinking of fungi as mushrooms. Mushroom is the fruiting body of fungi. It's also called a sporocarp. It holds the fungal spores. Mushrooms aren't the only type of sporocarp. There are a couple different types of sporocarps. But when those spores are released into the soil, they germinate into something called hyphae. And hyphae are these thin thread-like structures that are very active below ground. So when we think about fungi and decomposition, hyphae are doing a lot of decomposition there below ground. But hyphae are very, very fine. They're 1 to 3 micrometers in diameter. And so that's finer than a piece of hair, like a piece of hair on your head. And they form these dense mats that sometimes ecologists that we call mycelia, or the mycelial network, because there's just these dense networks of hyphae below ground. And I love this picture down here because this is a dense mat of fungal hyphae on a leaf, and they're actively decaying that leaf. So for the purposes of the talk today, I'm going to talk about 2 groups of fungi that I think are probably the most important for dahlias. I'll be very frank, and I told this to Tony on the phone. I don't really know anything about dahlias. I had a great time listening to everyone talk right when I jumped on the Zoom. But I do know about these 2 groups of fungi, and I think that they're going to be the most important for dahlias. The first are the saprotrophic fungi. These are the decomposers. And the second group that I'll talk about in a few slides are plant mutualists, known as arbuscular mycorrhizal fungi, or AM fungi. And I'll talk about those in a few slides. But first, I'm going to talk about the saprotrophs. I absolutely love saprotrophs. I think decomposition, I'm biased when I say this, but I think it's the most important process in a forest. And that's because decomposition takes nutrients that are bound in once living organisms, like this once tree that's now a decaying log. It takes those nutrients and cycles them back into the soil. It brings them back to the soil. And so we call this decaying matter that used to be alive. We call this organic matter. It was once alive, now it's being decayed and being turned back into the soil. And so you can see on this decaying log here, there's a couple little orange dots. Those are mushrooms in the genus Mycena. That's a pretty common decomposer up in our forest at Holden. This picture up here on the top right, these are a type of sporocarp that are called puffballs. These are in the genus Lycoperdon. If you ever see these on a hike, they're really fun because if you touch them, they will explode and the spores will just release into the air. So I think it's really fun. So do my kids. But you see that these puffballs are covering this decaying log here. I also love this picture too. This is a saprotroph in the genus Gymnopus, and it's again decomposing this log that they're all covering. And so now I want to talk a little bit about this campaign. I love this campaign. It's through the USDA in Oregon. They are challenging landowners and farmers to soil their undies. And so what you can do is you go to the store and you buy a pair of cotton underwear. It has to be full 100% cotton, and bury it in the ground. And then you come back 60 days later and you dig it up and you see how much of the underwear has been decomposed. And so this gives an idea of how much decomposition is going on in the soils on your property. So, you too can soil your undies this weekend. But that brings me to a study that we did in the forest at Holden. We were estimating some of the fungi at one of our forests known as Stebbins Gulch. And we've been examining the fungi up there in Stebbins Gulch 4 times throughout the year: March, June, September, and December, so that we catch the changing of the seasons. And we've been doing this since 2006. So we've been examining the fungi over the course of all 4 seasons, dating all the way back to 2006. And what I can tell you is that even in the winter, the fungi are quite active. It's cold for them, but they are still performing a lot of decomposition all year long. And so here's a picture of one of our interns. This is Rachel Reeb. She's now a graduate student at the University of Pittsburgh, but when she did an internship with us, she of course helped us collect soil up there in Stebbins Gulch. And to orient you to where Stebbins Gulch is, Holden Arboretum, we're in northeastern Ohio, about 20 miles east of Cleveland. So right here near Lake Erie. And then this is an aerial photograph of that forest. It's a really nice, fairly intact forest with a little bit of development around it, but it's about as natural as you're going to get in northeastern Ohio. And as I mentioned, we collect soil and examine the fungi at 4 times throughout the year. And so that means that we're up there on days that look like this, like in the first picture I was showing you where there's just wildflowers everywhere. I showed you spring beauty and trillium. This is that squirrel corn. This is purple violet, and then that wild leek that just really carpets the floor. But we're also up there examining fungi on days that look like this, where it's very snowy and I'm wearing my wool hat. And as I mentioned, the fungi still are really active below the soil, below the snow. And so we hypothesized and we thought this change in temperature, these changes of the seasons, should really matter for the fungi. In fact, these are some data that we published in the journal Ecosphere, and the top graph is showing soil temperature at just below the surface, so only 2 centimeters depth, so just below the soil surface. And what you can see, and it's not surprising, the soil is cold in the winter and warm in the summer, and this cycles throughout each year that we have been doing this study. What I'm showing here are the first 5 years, so from 2006 to 2011. And then when we look at the variation in temperature, the daily variation in temperature, we see these periods where there's pretty high variation in temperature. So you see these dots on the graph that are kind of up near 8 or 10. And this is in the springtime. And so we all know that there are days in the spring where we leave in the morning and we're wearing our winter jacket. And then by 3 o'clock, we're in just a t-shirt and maybe we put on a pair of shorts because it's much hotter. And so that's what those periods of high variability are showing. But then we also have these periods shown with these blue brackets here, where the daily variability in soil temperature was really low. So the temperature was very stable over the course of a day. And these periods correspond to snow cover. So where Holden is, we're in the snow belt. We get a lot of snow typically, and it also typically persists for several months. So we have these time periods where there's not a lot of variability in temperature. It's cold, of course, because it's winter, but the temperature is not really changing. And so we thought, okay, this has to really matter for those saprotrophs. This is really what's gonna matter for the decomposers. It turns out we were wrong. Our hypothesis was not correct. This is another graph from that paper we published in Ecosphere. And the bars that are here in the shades of red, that with the orange shade behind it, are all measures of temperature and how influential they were for the saprotrophic fungi. And you see how these bars are all below 1? That says they were not very influential at all. The things that mattered for the saprotrophs were measures of soil nutrients or soil chemistry, things like the phosphorus that was there, the nitrogen that was there, the carbon, and the water content. And so what we've really concluded is that events like this is a tree that came down at one of our sites. And so we know it is because we have this laundry basket here, this very scientific laundry basket. We use the laundry baskets to actually collect the leaves when they fall in the autumn, and we measure the amount of nutrients that are being put back into the soil. At any rate, this tree fall was a much more significant event for the decomposers than any of those temperature changes, whether they were seasonal temperature changes or daily temperature changes. And this is something that scientists have found in other forests as well. There's a group in Hungary that work with fungi in the boreal forest there, and they are finding very similar things. These really kind of small events that change just a really small portion of the soil matter more to the fungi. And so then really the take home from this is that what matters the most for our soils at home is the inputs of this organic matter, this stuff that was once alive, that the decomposers can then go in and decompose and cycle those nutrients back into the soil. So something like a wood mulch, I know we lay, at my house, we lay wood mulch every year, or leaves or compost or manure. These are all really good for the saprotrophic fungi. These sorts of organic inputs are really important for the plants because they feed the fungi. And then the fungi in turn do all the decomposition and release those nutrients back for the plants. So that's what I wanted to say about saprotrophs. Now I'm going to talk a little bit about mycorrhizal fungi. Mycorrhizal fungi are plant mutualists. They live in association with the plant roots. And they exist in what's called a mutualism. So that means that both parties benefit. So both the plant benefits and the mycorrhizal fungi benefit. And so what happens is, I love this picture. This is from a book called Mycorrhizal Symbiosis by Smith and Reed. And you can see this pine seedling here and the taproot that's extending down into the soil. And you can see all of the fine roots coming off of the taproot. But then this white haze that's behind the fine roots and that's extending from the fine roots, that's all fungal hyphae. And so what's happening here is that the mycorrhizal fungi, they're associating with the plant root, and then they're extending their hyphae beyond the roots, and they're acting sort of as they're acting as an extension of the plant roots. And what happens is the mycorrhizal fungi, they acquire nitrogen and phosphorus from the soil, and they give some of that to the plant. They're not doing that out of the kindness of their little fungal hearts. They're doing this in exchange for carbon from the plant. So the plant fixes carbon during photosynthesis, it makes sugars. And so it takes some of those sugars and gives some of them to the mycorrhizal fungi in exchange for the nutrients from the soil. So this is why it's a mutualism. Both parties are benefiting. The mycorrhizal fungi are getting carbon, and the plant is getting nutrients, especially nitrogen and phosphorus. And the type of There's a couple different kinds of mycorrhizal fungi, but the type I'm going to talk about are the arbuscular mycorrhizal fungi. These are the ones that dahlias associate with. And I have a couple papers that I'll show on the next slide. But arbuscular mycorrhizal fungi, they actually live inside the plant roots. And so you can't see them with the naked eye. You need to take the plant root into the lab and you need to smush it on a microscope slide, and then you need to stain it with a dye. And so that's what these two pictures here are showing. They're showing plant root cells under a microscope, and you can make out some of the cell walls. So, plant cells are rectangular in shape, and so you can kind of make out some of the plant cells there and in here as well. But as I mentioned, these have been smushed, so some of the cells do get disrupted. And then the anything that you see that's in this top picture looks blue, or in this bottom picture looks sort of reddish, like a deep red, those are fungal structures. So those are structures of the arbuscular mycorrhizal fungi. These ovals are called vesicles. Those are storage organs. So the mycorrhizal fungi are storing a lot of their nutrients there. And then these structures that sort of look puffy, sometimes people say they look like bubble gum. These are what are called arbuscules, and they're actually really structures that look very branched. That's how the AM fungi got their name, because they arb for tree, because they look like a branched tree within the cell. And this is where all of the nutrient transfer is taking place. It's within these arbuscules. So this is where the plant is giving the fungi carbon, and where the fungi is giving the plant the nutrients. And then you see these stringy structures that are sort of emanating out of the root. That's that fungal hyphae that's going to extend out into the soil and grab the nutrients for the plant. Now, nutrients is not the only benefit that AM fungi have for plants. They also help during drought, and they also help resist disease. And we think that's because they're a physical barrier. So they're this physical presence that can prevent other pathogens. I shouldn't say other pathogens, but that can prevent pathogens from getting into the root, or can help that root from drying out during a drought. And there are a number of plants that associate with AM fungi. A lot of herbaceous wildflowers, like I keep talking about, like trout lily and trillium. This one's wake robin here, and wild leek, as well as a number of tree species, such as the maples, both red and sugar maple, and tulip poplar. Tulip poplar's in the magnolia family, but we find them a lot in our forests. And I actually think that's what makes forests in Ohio really unique, because I'll hear scientists say, well, trees don't associate with AM fungi, they associate with other groups of fungi. And I'll say, well, maybe the trees in your forest don't, but the trees in our forests do. The maples, the tulip poplars, black cherries. Black cherries are a little bit more minor component of our forest, but still we find them around. And sorry, that was kind of a side note, but it's something that I tell scientists all the time, because I think it makes Ohio pretty cool. And as I mentioned, turns out these are the fungi that dahlias are associating with. When I was doing some research for this talk, I came across this paper by a group in India, and they were assessing the degree of colonization in a number of ornamental flowering plant species, one of which was a dahlia. And the dahlia was just lousy with AM fungi. It had 88% colonization. So what that means is that when they were looking under the microscope slide at the roots, they would take a field of view, and then they would say, okay, yes, this field of view has mycorrhizal fungi, or it doesn't. And so when they did that, out of 100 times of them assessing whether or not the root segment had AM fungi, 88 times it had mycorrhizal fungi. So, and that's really high. I can tell you that it's lousy with AM fungi. I also found this paper. This is from a group in Romania. They were also assessing root colonization in 10 species in the Asteraceae. So Asteraceae are known to associate with AM fungi. And I'm not saying anything that you all didn't know, but it was new to me, that dahlia are in the Asteraceae family. And this group, they themselves didn't measure the colonization in dahlia, but they were comparing some of the Asteraceae's colonization in the species that they did measure with other measures of dahlia that someone in Romania had done and just hadn't published. So anyway, I thought that was kind of cool that I found some of this stuff. But I will tell you about some of our work with AM fungi in our forest. And so we've assessed AM colonization in wild leek. And so I've shown you a few pictures of the wild leek in our forest, and there is just a lot of it. It just really covers the forest floor throughout the springtime. And so we thought, well, these wild leek have to be really important for the nutrients throughout the forest soil. So let's take a look at them. And so we did a couple different things. One of the things we did is we measured the nutrient content in the bulb of the leek. So the bulb is their storage organ, probably similar to the tuber. Although, you know, someone can correct me if I'm wrong here. I was reading about dahlia that the tuber botanically is really a root. Is that, I don't know if I'm correct there. So sometimes I saw it referred to as a tuberous root, but functionally it's a tuber, right? It's a resting tuber. I don't know. Does someone have anything they wanted to add to that?
It is a tuberous root. It's a thickening of the root.
That's so cool. So sorry, I just, you know, as you could tell, I did some research for this and I found it all just really fascinating. Okay, so anyway, back to wild leek and AM fungi. We measured the phosphorus content in the wild leek bulb, which is their storage organ, as well as colonization by AM fungi throughout the 1 year of the wild leek life cycle, from its leaf out through flowering and seed set into dormancy, and then through the emergence and elongation the following year. And what we found is that when the wild leek had emerged, between that emergence and the elongation here at the end of the graph, we saw this pretty dramatic decline in the phosphorus content of that bulb. So, you know, this is a really costly thing for this plant. You know, growth it's an energy cost for the plant. So it's probably not surprising that the plant emerges and it grows and then it uses the phosphorus that it had stored up. And this decline in phosphorus in the bulb coincided with an increase in colonization by AM fungi. So one way that you can look at these data is you can say, okay, well, we know that growing in the spring is really costly. So if we were going to apply fertilizer, we would do that, you know, just before emergence, sometime in the early spring. But another way to look at these data and how I would interpret them is that you don't necessarily need to add fertilizer if you've fostered the right environment for AM fungi, because the plants will be able to cope with that decline in phosphorus by associating with their mycorrhizal mutualists. I do want to mention that wild leek, they have a perennial root system, and we've done some work comparing species that have perennial roots. So these here are called false Solomon seal. They are a wildflower species that has perennial root system. And we compared these with Jack in the Pulpit. Jack in the Pulpit have an annual root system. And so I believe dahlia have an annual root system. Am I correct there? I don't know.
I would say so, yes.
Yeah, they have an annual root system. Okay, so this was some work we did with these two species. And what we found was when the Jack in the Pulpit, which has the annual root system, was colonized by AM fungi, that's this plant right here, we had some increase in growth. I should say this was not statistically significant, but we also measured nutrient content, and they had significantly higher nutrient content within their tissue when they had access to mycorrhizal fungi. We didn't really see any differences in false Solomon seal. Our hypothesis is that that has to do with the fact that their roots are perennial and they maybe don't need the mycorrhizal association to the same extent. This was one case study, just two species. But anyway, the take home is that whether it's an annual or perennial root system could matter. And hopefully I've convinced you that mycorrhizal fungi are important to have in your backyard. So what are some things that we can do to foster mycorrhizal fungi just right in your own backyard? So one thing is plowing and tilling can actually disrupt what's called the fungal mycelia. That's the fungal hyphae that network below ground. And so we actually would recommend no-till gardening practices if fungal mycelia is something that you're interested in, if these AM fungi is something you're interested in fostering. It will keep the mycelia intact, but of course, no-till gardening also has some other benefits in that it can reduce erosion of the soil, can increase water content of the soil. Adding fertilizer can actually reduce mycorrhizal colonization. So we would recommend inputs of other types of fertilizer, like organic slow release fertilizers. Things like manure and compost work really well. And these will increase the spore numbers of the mycorrhizal fungi. They also will provide organic sources of nutrients, which is a little bit more natural for them. Cover crops also work really well in that they'll keep the mycorrhizal fungi intact over the winter. I was talking to one of my colleagues, and he really recommends like a cold weather legume, because not only does that legume associate with AM fungi, they also associate with nitrogen fixing bacteria. And so you're fostering both AM fungi in the soil and also these nitrogen fixers, which can increase nitrogen in the soil as well. And finally, pesticides can harm mycorrhizae. So we would recommend using pesticides sparingly. Not saying to not use it at all. I use pesticides in my yard too. I've got some poison ivy back there that I always react to. So I'm not saying to not use it, but I'm going back there and I'm spot spraying it. I'm not doing a blanket spray. So those would be our recommendations for fostering mycorrhizal fungi in your own backyard. And then the last thing that I want to talk about is some other work that we had done in our forests where we see really large differences in the soil. So you can see just from looking at these soil profiles that they look quite different. This one here on the left is just, one color of soil. It's pretty much clay all the way throughout. And it turns out that this soil core, though it was taken from a forest that exists today, it wasn't always a forest. Based on some aerial photographs from 1937, the forest that's there today was a barnyard back in 1937. So it was under agriculture at that time. And so if we compare this soil profile with the one on the right, which is from an old growth forest, we see that in our old growth forest, this really nice layer of topsoil here at the top. And this is just from a lot of decomposition of organic inputs over many, many years. And the fact that the soil hasn't been disrupted, say by tilling or by planting of an agricultural field. And we know that these forests that here I'm calling high quality, we know that these are old because we had some tree ring analysis done by the College of Worcester a couple years ago, and the larger trees are dating back into the 1700s. We had one that dated back to 1760. So these are really old forests. And so of course, because we love fungi, we had to take a look at the fungi below ground, and we do see some pretty big differences. I'm going to start with the blue circles here. Which are the saprotrophs. And what we find is that when we looked at the fungi in these two situations, in a forest that used to be an agricultural field and in a forest that's really old, they have the same amount of saprotrophs. 20% of the fungi that we found saprotrophic. So that's good news. Lots of decomposers regardless of the forest history. But when we looked at the mutualists, those mycorrhizal fungi, we saw a significant difference. In our forests that used to have agricultural land use, only 5% of the fungi were mycorrhizal fungi, compared to 20% in our old growth forests. And as I mentioned, this was statistically significant, this difference. We also see a statistically significant difference in the pathogens. They're not as abundant as the saprotrophs or the mycorrhizal fungi. Only 1% of the fungi in our forests that used to be agricultural fields came back as potentially pathogenic, but this was significantly more than what we find in an old growth situation. Okay, so what do these differences mean? Well, for us, we do quite a bit of restoration work in our forests. This top picture here on the left, this is Acacia Reservation. It's part of the Cleveland Metro Parks. And we have done some fungal work with them over there. So Acacia used to be a golf course. And what they're doing is they're planting trees along the fairway. You can see this is a tulip poplar right here. So this is one of the trees that form those associations with AM fungi. And the idea is to try to get this old fairway to be restored into a forest. We're also doing some restoration in our forest out at Holden, where we are planting wildflowers just directly into the forests. But when we do these plantings of both of the trees at Acacia and wildflowers up in our forests at Holden, we're actually amending the soil. And we're amending the soil in 2 ways. We're either adding something called mycogrow. This is commercially available AM fungi. You can buy it at the garden store. So it's just dried AM spores. Or we are adding some really high quality forest soil from our old growth forests. And I can tell you, this is still a little preliminary, but I actually don't recommend this mycogrow. It seems like a great idea, but we actually are finding that it's reducing the growth of the plants. And so that's what these graphs here are showing. The plant species, the wildflowers that we had planted out in Stebbins, or sorry, I should say in the forest at Holden, are Jack in the Pulpit and false Solomon seal, the same 2 that we had done that study on, that I talked about. And what we're finding is when we add the commercial inoculum, shown here by the orange circles, the growth is lower. So we measured, for example, the corm of the Jack in the Pulpit, that's their storage organ, and it had a lower weight or a lower biomass compared to plants that did not have that commercial inoculum added. Same with the rhizome of false Solomon seal. So false Solomon seal, their storage organ is a rhizome. And when we add the commercial inoculum, that rhizome just weighs less, the biomass is less. This was also true for the leaf area of both plant species and the stem height of both plant species. So we think when we add that commercial inoculum, the plants are just not growing to the same extent. I have 2 hypotheses for why I think that's happening. I think either the commercial inoculum it's a one size fits all. So it may contain species of AM fungi that just don't associate with the plant species that we are using in our restoration. And therefore they're not going to confer any benefits because they're not going to associate with those plants. Or it may be that it's actually working too well because AM fungi, they are a cost to the plant. The plant has to give some carbon to the fungi. And so it's possible that we are adding just such a high concentration of AM fungi that it's becoming more costly to the plant than any of the benefits that come with it. Based on some of the data from Acacia, I think it's the latter, although follow-up studies need to confirm that. So the short of the long is I don't recommend the commercial inoculum. I do recommend using forest soil. We're seeing really good, we're not seeing this reduction in growth when we add the forest soil. And that, the species of AM fungi that are found in the forest soil are going to be adapted to the species that we are using during the restorations. And they're going to be in a nice concentration as well. So then just to kind of go back to some of the recommendations that I've been sharing, fungi in the soil, they like it messy, like this picture up here in the top left. They like a lot of organic matter. They like a lot of debris that's coming down into the soil for them to decompose. And so something that you can do at home, and this is on my red maple that's just behind me in my backyard, we do mulch around the base of it. We don't volcano mulch. Volcano mulching can actually rot out the bottom of a tree, but a thin layer of mulch about 1 meter out can create a more natural environment for the tree. And my husband likes this because he thinks it looks pretty. So that's fine. If that's why he thinks we're doing it, that's great. For me, I'm doing it so that I can foster those mycorrhizal fungi below ground and create a more natural environment than a lawn. And so again, things like additions of wood mulch or leaf litter. I heard a lot of people talking about additions of leaves to your flower beds. We recommend this as well for the fungi. And then I'm just going to end by thanking David Burke is the principal investigator of the lab that I'm in. So all of the work that I presented today is stuff that he and I have been working on for, oh gosh, we've been working together for 15 years now. I need to thank a really fantastic group of volunteers. They call themselves the Research Renegades. They come in and help us with our research projects, especially that restoration work that I was talking about, they did a lot of those plant measurements. You can see the group here measuring the Jack in the Pulpinum, the false solemn, and seal for us. They're a really fantastic group. I also need to thank a lot of summer interns. They help us to collect a lot of data. And I always like to end with this picture. These are my children. We got all dressed up one day to take family photos, and they of course had to play in the soil. And so I like to tell everyone, you are never too old to get down and play in the soil. All right, and with that, I'd be happy to take questions. I also would be happy to learn anything about dahlias. They just sound fascinating.
We do have one question from
Sure.
Bob. He wants to know, does the golf course have carry over elements from the previous use?
Yeah, I'm sure it does. You know, one of the things one of the reasons why I wanted to show those two soil cores of an old agricultural field and an old growth forest is, you know, what we've done in the past really matters for today. And our data suggests that even, you know, 80 years of forest regrowth is just not enough to bring back the fungi that are there. Did I answer your question, Bob? He was interested more in debris or in contamination, soil contamination.
Influencing your data on the plants growing that you recorded. You know,
Oh,
some of the harsh chemicals on a golf course to make it nice and green or keep the weeds down. I just wonder if those trace elements would have retarded the growth of the plants that you were measuring.
oh yeah. So so when we were involved, we were measuring just the fungi, right? I do know that when they were establishing those plants, they didn't really have a problem with getting them established. That's a great question as to how well they've done since then, because we were just looking at the fungi like kind of early on. But I think you're right. I think any of the chemicals that were used back then could have an effect long term. My inclination is that they selected species that they think would be hardy in those conditions. Like I know tulip poplar, for example, is a really hardy early successional species. So if you were to like leave a field abandoned, it's one of the first species that's going to start growing. So my guess is that's why they picked that one. They also picked black cherry probably for the same reason. Yeah.
And Misty has a photo that she wants to share. Give me 1 second. Can you guys see this? And Misty, do you want to describe what?
Yeah, so thank you so much for the presentation. I found it really interesting and learning lots. I think this is mycelium. Is that?
Yes, it looks like it for sure.
Yeah, around my dahlias. So I've been noticing this and thought it was interesting that, you know, I have it in some of my garden beds.
Yeah.
When I dig up the tubers. So I'm really curious as I'm learning about like mycorrhizae and stuff. I actually have used you know, what is that? Like an inoculant.
Yeah,
I put it around. Yeah, I put it around, but it sounds like maybe you don't need that per se. So, but I have been doing it. And I don't know if that's, you know, reason why I'm seeing that, or I would maybe see it eventually.
Yeah. So yeah, potentially. So I think one of the things that's difficult for me to tell from the picture is whether it's a decomposer that's in there or whether it's mycorrhizal. So if it's directly attached to the root, then you know that it's mycorrhizal. And so it's like emanating from the root.
Yeah, it seemed to like kind of surround itself around the tubers.
Yeah, it looks really cool. Sorry.
Yeah,
it looks really, really cool. Yeah, so but I would say you probably don't. So I mean, if you're adding the commercially available mycorrhizal fungi, I'm sure that it's going to confer benefits. So you know, but you don't have to. You can just use what's in the soil. And someone had asked a question on the chat. It looked like, how do you get forest soil? You can just right away and ask for a permit. So when we add the forest soil, we're only adding a gram in the planting. It's not a lot. It fits in, I mean, it fits inside a really tiny Ziploc bag. So if you were to write away to a permit to the Metroparks and say you just wanted to collect some soil, this is what I wanted to collect it for, or even to hold it. And we have a conservation department that grants permits, and just be very specific to say, I'm only going to collect, you know, a couple shovels full of soil. It's a very small amount because we're not trying to change the overall soil. We're just trying to add in a little bit of microorganisms.
So I do have another question, actually. Sure. So if essentially you're trying to recreate what's happening in the forest, like in your own garden, mm-hmm. Is there a test that can test what the, you know, fungus, you know, soil, because it seems like generic soil tests don't test that. No. And where could you find that? Because it seems like the generic soil tests will tell you what your NPK and all that kind of is. So then human nature is to then go buy those commercially available, you know,
right,
amendments to come in and do that, instead of saying, well, hey, maybe it's like a fungus test that you want, and then create more of that natural decomposition, which would make those nutrients available.
Right. So did you see my slide where I said about how you can soil your undies? That's the best way to look for decomposers, is to bury something and then just see how much of it is decomposed after 60 days. And so that can't be, you know, you do need 100% cotton underwear. Now, the mycorrhizal fungi, that's going to be a little bit different. Based on the picture, I do think you have some mycorrhizal fungi in there, but they're going to be a lot harder to tell. I could tell you what we do in the lab is we do DNA sequencing, and that's how we find whether or not they're there. And that's not something that's going to be available to everybody, right? So I don't know if there's like a really good and easy commercially available way to look for mycorrhizae, but there are things you could do to help foster them in your soils below ground, right? And so let's say you do measure NP and K. You measure them this year, and then you bring in some compost and you add it to the top, and then you measure it again next year. If it looks like it's increased, then you know that your practices are having an effect. So I guess that's one thing you could do.
I had a question about companion plantings. I know there's been some research done on plants grown with other plants, and even in some orchards where the trees do best if they have cover to the soil around the trees, rather than like we grow our dahlias, most of us, pretty sterilely. Almost every plant, no weeds. We don't want any weeds or anything like that. I can remember 40 some years ago going to a dahlia garden, and the entire dahlias were ground covered with portulaca, the moss rose. The dahlias were doing great. And I've often wondered if it's actually beneficial to have other plants growing in between them because of the mycorrhizal network and the sharing of nutrients and just beneficial effects that roots have on each other, rather than just relying on our dahlias every 3 feet. And dahlia roots will go out 3 feet from a plant, so they will kind of intertwine, even though we can't see them. But I just wondered if you had any thoughts on that.
Yeah, so I'm sorry that I'm going to bring it back to wildflowers again, but that's what I know. And so the study that I was talking about with Jack in the Pulpit and False Solomon Seal, the annual root system versus the perennial root system, for that study, what we had done is we did co-plant them. And so we had the Jack in the Pulpit in little tiny seedlings in the same pot as what we called a parent seedling. So they were in with a 3-year-old seedling of Jack in the Pulpit. And then they tapped into the same mycelial network as that parent plant. And then that's what conferred the benefit to them, is they had to have that parent plant there. Otherwise, they wouldn't have no benefits of the mycorrhizal fungi, because the parent plant already had the network set up and then they just grew right into it. So at least in a natural setting, I would agree with that. Yeah.
I've often thought that just nature doesn't leave stuff bare in between plants. And so it's probably not the best.
No. And, you know, the other thing is a lot of times plant species do better with an opposite species, because it's almost as if whatever signaling goes on below ground, they know, hey, this isn't the same species. I need to do better. I need to compete better, if that makes sense. In fact, I had a professor when I was at Case he studied animal behavior, and he used to say to me, I think plants do everything animals do. They just do it slower. Oh, okay. There's no annual roots compared to perennial roots. Tony will have perennial roots for the second year growth. So they have a perennial root system?
I think Tony earlier before this started mentioned that he had some dahlias that overwintered. And I think, Bob, I'm not quite sure what you're asking.
Yeah.
So you showed a slide where you're showing annual roots and perennial roots, and we came to the conclusion that dahlias were annuals.
Yes.
And they have to be because we dig them out every year. So
I
they're
see.
one season. Now, Tony mentioned earlier that he had left 3 in the ground and they'd already sprouted. So they're going to have like a second season of growth. I'm just wondering if there's going to be a different root system coming out of those dahlias that we've never really explored.
Well, I mean, I know people in the Pacific Northwest overwinter their tubers. I don't know that they're any different, right, Tony? They're just massive.
Yeah, I think they dig them a lot of people dig them every other year. I think, Christine had a clump that she'd left in the ground that she had just recently on Instagram that she left in the ground for 5 years. And it was about 3 feet in diameter, and it was massive. And there was a lot of rot on it. So it's not the ideal way to grow them for production, is to leave them in. They really benefit from being divided, but they will, in warmer climates, they'll just keep coming back, but with multiple shoots. The eyes are just enormous on some of these varieties.
And I think some of the people out there leave them in, but dig them and replant them in the spring to divide them. So that's almost what we do.
Yeah, some people dig them yearly, some, every other year. I think everybody digs them at least every other year.
Sarah, a completely different question. Where do your students come from that help you during the summer?
So our summer interns come from all over. We tend to advertise at universities, but it's also a publicly available application. So let's see, we've had students from University of Michigan. We've had students from Syracuse. We've had students from West Virginia University, and then we get quite a few local students too. So in fact, our summer intern, her name's Alexa. She's starting on Monday, and she's from this area, but she's coming home from the summer. But I can't remember now off the top of my head which university she's from. But I don't know. It's so much fun because to get to be around young people who are so excited about science, it's just so much fun.
I was just curious. I teach at John Carroll, and I was wondering if you work with any of the botanists there.
So we know Rebecca Dranowski. She's come out before with her class, and we've done some demos with her class before.
Okay.
And I remember hearing her speak at Case too when I was at Case. So yeah, we've done some stuff with Rebecca. In fact, we have a graduate student now who did her master's at John Carroll, and so now she's doing her PhD. So Holden doesn't, I mean, for us to have PhD students, they're like half-time Holden students and then half-time through Case. So technically they're Case Western Reserve University students, but Holden like pays half their salary, if that makes sense. But Sharon did her master's with John Carroll, and then now she's with us for PhD. So it's, I don't know, it's very exciting. Summers are my most favorite time at Holden, just to get to be around young people and get to talk about science. And I had the student from Gilmore who shadowed me the past 2 weeks, and I've never had that. It's never happened before. I never had someone ask me to shadow before, but boy, was that fun. That was just, and she wrote me a thank you note. It was just, I don't know, the whole experience was wonderful.
They keep you alive, that's for sure.
Yeah,
keep you on toes too.
Well, and not many. In fact, we had someone. His name was Sean. He was at St. Ignatius. He's actually coming and doing an internship with us this summer. He reached out to me 2 years ago, and he said, I'm really interested in plants. Can I come out and just learn more about plants? And you don't get that a lot, like most high school students aren't interested in plants, but boy, was that nice to have him come out too. That was really fun.
Wonderful.
Yeah. What kind of work do you do at John Carroll?
I teach statistics.
Oh, fun. Oh, I do statistics. See, I should call you because some days I want to bang my head against my computer. So
Noticing on your graphs, you have error bars on several of those graphs too.
yes,
Yeah,
we try to do things correctly, statistically correctly. So
Jerry's actually going to fill out an internship form for the summer.
we'd love to have you, Jerry. Yeah. I'll tell you what though. So I'm sitting here saying all this stuff about the interns, but boy, do I love working with our retired volunteers. They are fantastic. It is, and they have a lot of fun too. So they'll come and they'll measure plants with us. Then they'll go out for a drink afterward. Then we have to stay and they can go out and have a drink. It's just such a wonderful group. Yeah. And we have a group that comes in. They don't work with us. They work with our horticulture department. They come on Thursdays, and we hear them they'll be down near our greenhouses. So the labs are in the building where we work. The labs are upstairs and the greenhouses are downstairs along with what's called a head house. And that's where we do a lot of our plantings. It's a really messy area. Boy, we hear the dirt devils when they come in on Thursday morning. They are having a great time potting plants. Yeah.
Well, hopefully we'll be back out because we've been, except for the last 2 years, have sold tubers and plants at Holden.
Oh, at the plant sale?
Yeah, at the plant sale.
Oh, wonderful.
And it's usually the Friday, Saturday, and Sunday, and they put us there in the building there, which is kind of nice. But so hopefully we'll be back there next year.
Oh, that would be great. Yeah, we get as I mentioned when Tony called me, I didn't know anything about dahlias except for that they were beautiful. The plants that we get that people come to Holden a lot to see are the rhododendron, because we have that rhododendron collection in Madison, Ohio, and we have a rhododendron curator. That's what most people come to see.
Yeah.
And boy, are they, in fact, like I said, the interns, when they start next week, we do a big picnic out at that leach station in Madison, just so they can see all the rhododendron in bloom. Boy, and I'm sorry, I have to say, and azaleas in bloom. And it's just absolutely beautiful.
Yeah. Well, thanks, Sarah. Any other questions for Sarah? That was a wonderful presentation. Really interesting.
This was so fun to meet all of you and get to learn something new. Boy, was that fun.
Yeah. But we'll have to get you interested in dahlias, that's for sure.
For sure. I will tell you, based on what I've read about them, I may not be able to grow them until my kids are a little bit older. Because right now we're running to Timbuktu for track meets and choir concerts. And
Where do you live, Sarah?
We're in Broadview Heights. So
okay.
It's like the southernmost part of Cuyahoga County.
Yeah.
Yeah.
Okay.
That's where I am. But I showed you that picture of David Burke. So this is another little side note. We learned that David has the community garden plot next to Tony's.
Oh.
Yeah. So David, who I've been working with for 15 years. And so Tony, he now tells everyone, he says, oh, yeah, the garden plot next to me is this Tony Evangelista. Boy, does he have beautiful dahlias growing.
I have my seedling garden there, and I've known David for years. I didn't even know he worked at the Arboretum. We would talk about plants and growth and soil. And I said, boy, this guy really knows what he's talking about. And then I met Sarah and she told me about David and I found out that they work together. It's such a small world.
That's a small world for sure.
That's a small world.
If you ever wanted David to come and talk, David is a gardener. I'm an ecologist. David is a gardener. He loves to garden.
Oh, good to know. We'll put him on for next year, Tony.
Okay.
Okay, well, thanks, Sarah. I really do appreciate it, and we'll get you into dahlias, and then your life will be complete.
Yes, thanks, Sarah.
Okay, let's see. Where are we now? So a couple of things going on. We, obviously we just had a tuber and a plant sale. Sharon, can you bring us up to date on both perhaps?
Well, the tuber sale, I didn't prepare too much on. We sold a bunch of tubers and we still have a few bags to be picked up. You know, there's always a few. On the plant sale, we started with about 500 plants. We sold about 269 of them for $1,842. So that would leave 228 left or something close to that. All these numbers are approximate because they're moving faster than I can keep track. And then so 60 go to Tom. We've got 44 going to sales tomorrow. Jessica's taking some to her sale and Rob's taking them to Mobius. About 20 will go out to Miller. And 24 to Elkhart, we figured, but we could probably send a few more. So we're going to be down to about 80 plants. And when you think of, you know, all the unknowns that Mike had to, you know, try to guess what to grow, I think that's pretty exceptional. And everything looked really nice today when we looked at it.
Yeah, good.
Sharon, let me add a thing. Mike had separated Tom's and the Miller Arboretum off to the side before we counted. So he had picked out special ones for
Okay.
those two
So-
sites.
So
So
let's see.
That's probably 80 more onto the list instead of subtracting them,
Oh, okay.
because he wanted to give Tom pot roots so that we'd get them back next year for starting cuttings. And then he was planning on giving Dave Cap dahlias to Miller to kind of create a little garden on his behalf.
Yeah, and they were mostly Dave's tubers. I do know, or Dave's plants.
So I think when we counted on Tuesday, it was 152 plants were left, or 252. Jeez, 252.
Well, it didn't look like that many, but maybe.
Well, we sold, I
We'll see what
picked
we
up,
have after Sunday.
well, I picked up another 15 or so, 10 or 15.
Yeah, I did put those in my sold list.
You did, okay. So I'm wondering, of course, we don't have a lot of people here tonight. I was even going to suggest that those who, if we could get the list of the ones that are available.
Well, it's getting to where that would be a little bit flaky. I mean, we could say probably available or something like that.
Okay,
I mean, unless somebody goes out and just totally re-inventories, because
well, for those who come Sunday, I mean, we'll obviously mention to them that there are others available.
what about just putting them out and selling them for, I don't know, $5, $4?
Yeah, well, that's what I was
Even if you
thinking.
get $1, it at least they pay for themselves.
Yeah, that's what I was thinking. So at least for those who, but we only had, how many people actually bought this year?
I can tell you if you want to wait a second.
Okay, it seemed to me there were fewer than last year.
18 to 20 orders probably.
Well,
many?
last year it was open to the public too sooner. I think that's probably, we should have had it open to the public the whole time.
That's a thought. Especially if we do it this way again next year.
No.
Now, somebody will do it again next year. I think this really goes better than the auction, to tell you the truth. But we shall see.
Well, hopefully we'll have Holden and Rockefeller next year too.
Yeah. I see Mike's not with us, so tonight, but anyway, okay. Well, that's good. So what we did for those who who care, we moved all of the plants over to one of the greenhouses that wasn't as warm as it will be, particularly tomorrow, because the ventilation in the one that we grow in is not great. But pickup is Sunday, and all the ones hopefully everybody will pick up and buy some others, and we'll be okay. Then we'll have to decide for whatever is left after that, what to do with them. So keep, keep looking at your emails next week. Anything else? Jess, do you have anything to add to that? No, okay. Okay, well, thanks Sharon and Rob for the work that you did out there today with Mike. Did appreciate it.
2, 3, 4, 5, 6, 7, 8, 9, 10.
Randy and I and Bob Matusek are going to Brant Greer's barn tomorrow to take an inventory on the old vases that we have there. And we'll see how many there are. We'll see how many the mice may have chewed up. But at any rate, Randy hopefully will take some. Pittsburgh is needing vases for their Midwest show, not this year, but next year. So we thought we would just, as we voted last month, if you recall, to give them some, and we'll just see how many are there. And then I'd like to make them available to members too. Would like to have, 3 or 4, something to share with the flowers that they may cut and give to local libraries and so forth and so on. So we'll do that tomorrow and then let you know. Mike and I will also be visiting Mary next week sometime and plant those border dahlias that she really likes and Dave grew so well. And I saw the ones out that Mike started out at Willoway, and they really are neat.
They're blooming.
Go ahead.
They're blooming.
Yeah, they are blooming. We'll also, while we're there though, take inventory of what's in the garage and get a little bit better idea as to what kind of space would be needed to store them. And that's another topic, where we're going to store all of the vases and oases and so forth. Because Mary's been very kind and to let us, keep them there now, but I know she doesn't want them there forever. That's for sure. So if anybody has any suggestions on a barn or a extra garage or something like that that we could use to store our inventory with, that would be very much appreciated. Let's see. Jess, do you have anything on your list of things you want to let the membership know about?
I don't think so.
Let's see, we talked about, what did we talk about the other day I thought there was at least one thing you might want to share.
I think maybe the big thing that is going to change is Nancy and I are going to switch to using MailChimp completely. So instead of Nancy sending emails from her personal email address, we're going to use a system to send it out, because right now Nancy has to send 4 different emails because our membership has grown so much. So we're going to use a different system. We'll give members a heads up before that first email goes out. It may end up in your spam folder, or if you use Gmail, your promotions folder. So we'll send out a personal email to encourage you to look, just in case it doesn't show up in your regular email. That way you won't miss anything.
You want to say just a quick word about Storefront?
Oh, the other thing that we're looking into is developing a store where we sell DSO merchandise. So like store or t-shirts or sweatshirts, mugs. So we can put a logo, or I kind of mocked something up quickly using the header that I'm using in the emails. So we're looking into it. I ordered some samples to see what they might look like. But that may be in the near future as well. So if you want to order shirts to wear to like some of the shows, you could. But we'll keep you posted.
Great, thank you. Randy, a word about the upcoming Midwest Conference business meeting?
And in Southfield.
You're muted. There you go.
Yeah, well, things are on a roll. We're a little late on registrations. People are really dragging their feet as far as getting their registrations in, and in our times, that's understandable. Some people are afraid to make an early move, and that's fine. But I think it's a good time. We're going to have a panel discussion, and our very own Tony Evangelista has agreed to sit on it. Tony's going to cover most of our organic questions. I hope he's not inundated with that. But that said, everything's moving right along. And I have one question to ask, and I'm going to ask the general audience at the general meeting there is, with the help of Sharon and Jim Kasner and a few others that we put out the Midwest Diary Report and some other. I'm curious and excited and wondering, is it getting passed around to the members of the locals? And if it's not, then we got to figure out a way to do that. Sharon's invested in this far over what she ever wanted to be, I'm sure, but she's done a bang up job, and I hope all the members are enjoying that. But that's pretty much all. Anybody got any questions?
Great, thanks.
Silence, I love it.
That's the first Saturday in June in the High Dive Pavilion in Elkhart. For those
The auction is right, or
that's an auction, yeah. And it's a business meeting and then the auction. And Tony typically has been heading up or helping at least with the auction. And I'll be talking, well, okay. Let's see, one other thing was, I guess maybe Q&A from particularly for some of the newer people. Do you have any questions from when to put our plants or tubers in the ground? If we just put bare tubers in the ground, how deep do we put them, and so forth? No question is too simple. But
A lot of bashful people out there.
yeah, I think there's only 2 new members tonight, Kathy and Goldie.
How are the two of you doing with starting your dahlia? Have you started them inside, or are you just going to put them out, bare tubers out in the ground?
I've got all mine potted up and just waiting for the next few days of rain to go through. They're sprouting and looking good.
Great. How many are you going?
Oh, if I count them and total them, I feel like that's admitting my obsession. So I ordered 60 new ones this year, and I probably have 40 from 45 or 50 from last year. So I just like to pretend I don't know the total.
Yeah, I just keep hiding boxes as they come in, Kathy. So
Yes, yes.
he never needs to see them.
I just got notification of one last shipment that I forgot about, and I was like, oh, where are those gonna go? Order another raised bed.
Goldie, how about you?
Well, I've got mine planted, not outside, but inside, trying to get
Yeah.
them to sprout. I've got some that's like probably about 6 inches high,
Okay.
and some, every one of them has got something growing out of it. So I know they're all good.
Good.
So I think I'm in good shape on that. I'm just waiting for it to stop raining so I can put them in the ground and then I don't have them rot on me. And as far as my count, how about 66 this year?
Pretty
It's like,
good.
okay, like she said, obsession. My husband gave me 2 new raised beds this year, and I have 3 right now, and they are 4 by 10, and he wasn't able to get the 10 foot, so he had to get 12 foot, and I said, oh, bummers. So that gives me another 6 because I got 2 of them.
Yeah.
So I'm like pretty pleased with that. So it's going good. They all have some eyes that's growing, and that's what counts.
Yes.
And I,
Right now they're inside.
Yes, they're inside. I'm going to start hardening them off little
Yeah.
by little outside.
Yeah.
Some that don't have much growth at all are upstairs in a dome that I sprinkle water on there every other day.
Yeah.
But the ones that are actually getting more growth to it, I've got them in like 4 by 4s and trying to get them to grow with some, ground in that, soil. So I'm working on that, and I have grow lights downstairs in the basement, and I've gotten into a lot more money than I expected I'd get into. My husband's probably kind of ignore me about now. I'm enjoying it. Like they said, it is an obsession. You get into it, it's like, oh my. So
Wonderful. Gloria, where do you live?
Goldie, I live in the Kidron area. Do you know
Oh.
where Kidron is?
I do not.
Amish country.
Okay. In what county?
It's Wayne County.
Oh, in Wayne, down that way.
and I have
Do you know where
to,
Orville?
Yeah.
Yes, go ahead.
I have to thank Goldie because I had mentioned I was going to Amish country to Yoder's Produce where they sell soil and drip and all the connectors you need. And I needed to get another line of drip tape. And Goldie and I were chatting and I was telling her about these annuals that I always buy for the front of my house. And she's like, go next door to Yoder's. They have really cheap annuals. And sure enough, I went next door and the plants that I always buy up here Petites for like $7.49 were $2.75. I left that place with 4 flats of annuals. And I called Nick and I'm like, I'm just warning you, this is what I'm coming home with. But I was like, it's awesome. So Goldie, I thank you so much.
That's another obsession I got into this year because they get everything in like once or twice a week. And they do, this place is where greenhouses buy them from as plugs. And then they pick them up and then they put them in the greenhouse. And I got like eucalyptus plants. I got 13 because I'm not massive like they are. 13 plugs. I planted them in 4 by 4s. And when I went to look at it, I went to Mount Eaton Greenhouse and looked at theirs versus what mine is. They looked identically the same. And I paid $1 a piece. And then over there, it's $4 a piece.
Every darn.
I went to that place every Thursday for a month and a half. And I got the extras, whatever they have on hand.
Well, Goldie, I'll be fighting you next year every Thursday.
Yeah.
Together.
Well, see, I can't get too much at one time because I work outside the home full time. So it's like a moderation. Okay, I get a small handful, I get them all set up, make it work, and then get another handful. That's how I did it this year.
Yeah, that's probably smarter than me walking out with 4 flats.
What was the name of this greenhouse?
I think,
Spring Valley,
yeah,
is it Spring Valley Flowers?
Spring Valley.
yeah.
It's just next lane south of Yoder's Produce. When you go one place, you go to the other.
And I will say Yoder's.
And go ahead.
I was going to say Yoder's is where I bought my diatomaceous earth. And it comes in this huge bag. And then I bought some alfalfa pellets. I bought my surround kale and clay that Tony does there. So I feel well stocked up for bugs this summer. I'm gonna give him my best shot.
Jess, your
Another
sale.
plug.
Go ahead, Goldie.
Another plug for them too. We all probably know Sunny Meadows where Greta and what's his name? Sunny Meadows that does tubers. They have greenhouses just in Xenia area.
Yeah,
Okay, for them, they actually bring a lot of their tubers up to this Spring Valley and they grow them there to get cuttings off of it for them.
Oh,
And so like Jessica, when you were there, did you see all those greenhouses that you weren't in? That's where they're at.
yeah, I did see it was really impressive, like the setup there. I don't know what I would expected to see, but it was not that. I think I have some photos.
Yes, yes.
So they just had flats and flats of petunias. I'm a huge Proven Winners Super Tunia fan. That's what goes out front in my house because they just grow massive. So yeah, highly recommend a trip.
When I heard of Kidron, I thought of Lehman's or Lemons Hardware. Been down there several times.
Layman's Hardware.
Lemons, yeah.
Yeah, they're my relatives.
Oh, really?
My mother was a Lehman's, so I'm a Kidronite. We call us Kidronites.
Really? Wow.
I was a Nussbaum.
We used to do business with Kidron Electric.
That was my uncle. Paul
Yeah,
I miss
Newsquender
the
is my
furniture
uncle.
store.
They own
What's
the
that?
town.
I miss the furniture store, Nut Tree.
Nut tree, that would be another Nussbaum. Nut tree means Nussbaum.
Okay. Well, I'm sitting at a desk that I bought there 20 plus years ago and I still love it.
I got a kitchen table from them.
Goldie has the connections, guys.
It's actually a dining room table with chairs. It's beautiful.
I have another piece of information, Jerry. I found my 21 plant sale. We only had 23 people last year buy plants. We had 30 this year, and we've already taken in more money than last year.
oh, I'll be darned. I would have lost the farm on that one.
Yeah, we had 7 more people, so that was actually almost 25% more.
Wow, neat. I may pick up a few more though on Sunday. So
Well, see, and
Jess, you have your personal sale or something tomorrow.
Yes.
And what do you sell?
Well, I'm gonna sell 30 DSO tubers, hopefully.
Yeah.
But we also have friends coming over. She does veggies. So we have heirloom tomatoes and peppers and kale and herbs. And then I also grow cut flowers. So I have zinnias, celosia, amihi, some sunflowers, pincushions, basils that I use for cut flowers. You could eat them, I guess, too. And a few other random things.
And where do you live?
In Cleveland Heights. Sharon and Rob came by, and they saw the disaster of my driveway.
it didn't look that bad.
We had 12 yards of dirt dropped off, and I think there's maybe a yard left.
Wasn't bad. You started with that much.
Yeah, it's a little muddy out there right now.
Well, good luck. Hope you do well.
Yeah, thanks. Otherwise, I'm gonna have a lot of dahlias left at the house.
Yeah, well, we'll talk next week about how to get rid of them. Gloria or Ron? Any questions?
No, no questions. We're trying to find Spring Valley Nursery in Kidron and we can't find it anywhere.
So if you look up Yoder's Produce,
Yoder's.
It's directly next door to Yoder's.
Okay,
And it's got Fredericksburg address, not Kidron. I'm from Kidron. It's 20 minutes, 15 minutes south of me.
what city did you say?
Fredericksburg.
Fredericksburg, okay.
And they do not have phones or
No.
anything. So it's very low key. I had to wait for somebody to come around. I think I hit them at lunchtime.
Oh, no. When I get there, I just mosey around through there. They show up.
Yeah, I mean, I did poke my nose around and then
And
somebody
the
found me.
Amish do have cell phones.
Oh, there you go.
Okay, we found Yoder's Produce on Apple Creek Road in Fredericksburg, so I'm assuming that's it.
Yeah,
That's
it's the next driveway. Yep.
Cool, thank you.
One we go to.
And it's right now, I don't think they're getting much more in, so you got to get there soon. Because, you know, when they're out, they're out.
I will say that Fredericksburg Nursery, I went there after this place, also had a good selection of flowers.
Or even Mount Eaton Greenhouse.
Yeah, that one too. I stopped there. I had quite the trip, quite the wonderful Friday without Nick and the dogs. It was just me and flowers.
Such a life.
Yeah.
Jess, you want to say a word about our speakers for next month?
Yeah, it's Tom and Sarah. They run Grateful Gardeners. They have a pretty big Instagram following. They just moved to a new farm, so they're spending a lot of their time redoing like all their flower beds and getting greenhouse up. But Tom got a grant to grow dahlias hydroponically. And so he's going to kind of talk about that. He's done it before, and now I think this new greenhouse that they're building is part of that grant. So he's going to talk about what it's like to grow dahlias hydroponically. So
Oh, it sounds really interesting.
Yeah.
Yeah. And then in July, Susan Cowling from who's a master gardener in Lake County will talk to us on composting. And then at the end of July, in fact, 2 weeks after the meeting, Jess and Tony will be doing a demo on staging, so that people, particularly newcomers, will be able to know how to or what to expect in stage and how to cut their dahlias and then how to stage them for a show, whether it's a Cuyahoga Fair or our Summit Mall show in Fairlawn, or Geauga County show, or our Petite show in Oakwood, which is the beginning of October. So all of those were indicated in the Dahlia Digest that Jackie did a really wonderful job in putting together the digest. But we'll be sending out emails on each of those also as time goes on. Any other questions or comments or topics people would like to bring up?
Can I ask a question?
sure.
That lady had talked about the soil that's in the forest. Is that much better than the soil we have out here apparently? Tony, you probably have an answer for that.
Well, I think Sarah's point was, we talk a lot about inoculating your soil with, microorganisms, which includes bacteria and the fungi. So what she's saying is that stuff that's been in the forest for 100 years has really all the right microorganisms in it. And if you just take very, very small amounts and seed your garden with it or your plantings, you're going to inoculate it, and then that's going to multiply exponentially through your soil and put in what needs to be there if your conditions are right. And it's the same principle of like using vermipost, like worm compost or earthworm castings, and taking a handful of those or a small amount when you're planting your plants and mixing it in with the soil as you plant. What you're doing is you're inoculating, seeding that soil with beneficial stuff to enhance growth. I mean, she wasn't real big on the mycorrhizal fungi, and I understand that because it's so prevalent, in our soils anyway, if you've got a long-standing garden. But that's the principle of it. It's a way to get a really high quality inoculant to put in your garden. You don't, like you said, you don't need much of it. A couple shovelfuls, and a pinch in around each planting, and you're probably gonna, but that's assuming that your soil is good enough for the inoculant to take hold. So I think the most important thing to do is to make sure that you're building good, healthy soil that is a good home for that inoculant, if that makes sense.
I remember when I was a young girl, my mother had me, behind me is 7.5 acres of woods. And it's always been woods. So that's going to have very good soil apparently with trees that were rotting back there, that type of stuff, because it's a forest. Anyway, she had me go back there and get some soil, bring it back, but then she had me put it in the oven for like 2 hours on low heat to get rid of anything that's bad in it. Was she onto something, or was that something that's like an old wives' tale?
I used to do that when I first started gardening decades and decades ago, and I really think it's an old wives' tale. I think it's really contraindicated. I think the more we learn about soil and realize that it's a living organism. She showed her first slide and it had that tablespoon of soil heaped up, and she talked about how there were millions and millions of organisms in that teaspoon. When you put that in the oven at low heat, what you're essentially doing is killing a lot of those. Some are more heat resistant and they're going to recover, but you've totally destroyed that inoculant. I still to this day do not understand why people buy sterile soil to start seeds and seed starting mixes. I mean, if you look at nature and your marigolds, they go to seed and you don't deadhead them. The next year you come out and there's those seedlings all over the ground. Well, nobody sterilized that soil over the winter. I mean, a lot of seeds need the microbial action to break down the seed coat, and then those mycelial fungi, those mycorrhizals are there to help bring nutrients in immediately. So the whole thing about sterilizing soil is just contra-intuitive to me. I don't really understand it. I know it was popular. I can remember putting trays of soil in the oven, back 50 years ago. I would never do that today.
I wondered.
I know that the greenhouses, the commercial greenhouses, I don't know if they still do that now, but they used to, in between crops, a certain time of year, when I lived out in Olmsted Falls area, they had a lot of greenhouses around. You could get the smell in the air, and it was because they would come along after the plants are gone. They'd heap up like in rows, like little mountains in rows, and then they would pump hot steam in the ground. And what that would do is, if the insects that be in there, they'd climb to the highest point of that hill, and then they would die. And then they'd cool the place down, go along with a shovel and take that the very top of those mounds and get rid of the insects. And this, I actually heard that from somebody, because years ago, when I asked them, why do that smell, where is it coming from these greenhouses? But I haven't, of course, been around greenhouses for a long time. That was what that was for. These hot, for growing tomatoes and things like that for insects.
You gotta try putting that stuff in the oven. The whole house smells like dirt
Yeah,
or something. It's awful.
yeah.
Yeah.
Just the difference in two, if I go and take compost soil or some good soil I've had outside and I bring it down the basement and I start potting up my plants early in the spring, I'll get a lot of gnats in the basement and I'll have to put these fly traps around and it grabs the gnats, you know. But if I buy the stuff from the store, the potting mix and this and that, you will not get those little insects flying around your basement. That's what I've found out.
Those are fungus gnats, you know, they live on organic matter. They're really not harmful. I mean, I'm
Right.
assuming that's what you're getting.
Yeah, they're just a nuisance, that's all.
They're a nuisance.
Yeah.
Right.
Any other questions or comments? This has been very interesting. Misty, thanks so much for joining us from the East Coast.
Enjoyed the presentation, so
Yeah,
be
she was
part-
excellent.
love you. Thank
Yeah.
you.
Okay, if Tammy, you have anything?
No,
No, okay.
thank you.
John, how are things going in your neck of the woods?
I got all my plants in the ground. Some are up 1.5 feet.
Are they
Wow.
really?
Wow.
Oh.
My problem is that I store them over the winter in my basement.
Yeah,
And so they get started early. I put them all in pots and only 1 out of 80 pots didn't come up. So
see.
some are still in pots, but I've got 63 in the ground. And that's all the holes I've got. So they're all started.
Wow.
I want to say a word about Layman's Hardware. I was over there last week, and one of their displays they have dahlia tubers in bags. I think you've all seen them in hardware stores and whatnot, but they're all dried up and just terrible looking. I wonder how anybody can make a living selling dead tubers.
Yeah, they use those, they call them pot roots. They're real small and they're all, you look at the bags, they're kind of broken up and everything like that. And
Yeah.
if you don't grab them, you let them wait till the like the very end of May and June, they're all dried up and everything.
Absolutely.
Like that. But I've gotten a few varieties before in the past where some varieties that I liked or wanted to get, and I'll usually try and get them early on March or April, and then I'll have them down the basement, and I might even moisten them a little bit and to keep them from getting dried out. But yeah, that's the way they all are. They're all look like they're all busted up and everything. But it's funny, they still have an eye on them, and the center of the clump, and they will still grow, a lot of them, as long as you don't let them dry out too much or buy them too late.
Well, it seemed it was too late for me. Yeah, at least there.
Okay, anything else from anybody?
Oh, one other thing I
Bob.
found out in going through my dahlias in boxes and all that, and normally I store them with vermiculite and all that. Well, I
Yeah.
started this past fall, I started wrapping some in saran wrap,
Yeah.
and surprisingly, even if I had some clumps, and I had very few that failed on me, or maybe 1 or 2 that clumps that had a few tubers that rotted, but I clipped them off, but the rest of the tubers are okay, and I had eyes on them. So I'm going to wrap a few more with saran wrap this coming fall. So like Dave did. Yeah.
Yeah, I did it. Ron showed me his method, and that's how I did mine. And I had really, really good luck, maybe too good of luck, because I had so many leftovers. But yeah, They stored really great.
Well,
Do you
you'll
have to put them in a cooler? Like when you put them in Saran wrap, I had it in Saran wrap. Do you have to put them in like a cooler of some sort, so below 60 degrees, or how do you do that?
I tossed mine in a crawl space in my basement.
Yeah, I've got an old coal bin. Guy got a 1920s house and they have a coal bin from when they're coal fired. And I keep them in a cool corner in boxes and they stay pretty good.
Yeah, we tried an experiment this year. We took a couple based on the saran wrap thing and we, knocked the dirt off and got them reasonably dry. And we put them in freezer bags, Ziploc freezer bags, and then stuck them in a cardboard box, left them in the garage because it never gets freezing in the garage, but it stays cool. And they were all light up this spring. So that's another, another way to go, I guess.
Terrific. Lisa, I see you just joined us. Jess is recording. I don't know if you saw the presentation or not, but Jess has recorded the presentation and also our conversation thereafter. So it'll be made available on our website, Sharon?
No, I'll send out a link via email.
No,
But
okay.
Lisa and I were texting each other beforehand. So
Okay, oh, so she
Yeah.
knows. Okay, terrific, terrific. Lisa, do you have any questions?
I think you're muted, Lisa. Oh. Yeah, I'm not sure if she's stayed on or not.
Okay,
We could put, if you want, next to the program, we could say, it is recorded and contact just for information. I just don't think we want to put it right on the website. Do you agree, Jess?
Yeah.
very good. Okay, anything else? Well, thanks so much for joining us this evening, and we'll see you next month. And some of this, maybe we'll see you Sunday, picking up your plants. And let's see, nothing else going on, I don't think, between now and the next meeting, right?
If you have a dahlias of today, don't forget to ask for it. Jerry will have them.
Yeah.
Oh, good.
And the ones that aren't picked up on Sunday, I'll try to get them to you somehow. Very good.
All right.
Thanks again.
Bye, everyone.
Thanks, guys.
Hey, everybody.
Bye.
Take
night.
care.
Thank you.
Thanks, Jess.
Yeah.
And Tony, thank you.
Thank you, Jer.